The hippocampus is a brain area that's involved in learning and memory. And the hippocampus keeps track of time for time epochs that are behaviorally relevant. The hippocampus has time cells that encode for time for behaviorally relevant epochs. In addition, there exists stimulus time cells. And the stimulus time cells actually represent the stimulus sometime after the stimulus is presented. And this is important for a task that's called the delayed non-match to sample task, where one stimulus is presented, the animal has to wait for a few seconds, and then a second stimulus is presented. And if the second is different from the first one, the animal has to respond and get a reward. In this study, we went one step further. And we wanted to find out if there was a time cell that would respond to a binary decision, yes or no, this or that. We performed our calcium imaging dorsal CA1. That's an area of the hippocampus that it's in the dorsal area, in the superficial area. And it's really important that it receives a lot of information from the olfactory system. When we recorded from the CA1, the animals were doing what's called a go-no-go decision task. In this task, the animals were going to respond to one odorant, if it was the rewarded odorant, and they would get water. We decoded the odorant valence, and we asked whether the cells that responded to the odorant valence responded differentially at different times. For example, when you're driving home, you're thinking, do I want to stop in the supermarket to buy some milk, or do I want to go home? And depending on the time and the situation, you decide to do this or that.
The go-no-go task is a classical behavioral paradigm that assesses mouse ability to differentiate and link selectively to specific odors in associative learning. Together with the ensemble activity recording of the dorsal CA1 neurons, using the genetically encoded calcium indicator GCAMP, we were able to collect valuable data to explore how the dorsal CA1 neurons encode temporal aspects of olfactory information during decision making. By analyzing the data, we found that animals respond differentially to the reward and unrewarded odors in associative learning, and the stimulus divergent responses of the stratopyramidal cells are largely heterogeneous, and the divergence increased with learning. In decoding analysis, we show that the activity of SP cells encodes the stimuli, and the learning induces improvements in odor decoding accuracy. We also found that the majority of the SP cells responds to the odor valence as opposed to odor identity when a reward is switched, suggesting the plasticity of dorsal CA1 SP cells.
Machine learning decoding of SP cells signals in CA1 involves computer training to interpret calcium signals, enabling the translation of brain signals into understandable information. Besides, decoding the stimulus from individual divergent SP cells revealed that there are tiling of neurons during the odor period, contributing to decoding, and decision-making took place at specific times after the odor stimulus. Leak prediction decoded from ensembles of SP cells correlated linearly with the leak behavior, indicating that sequential activity of the cells constitutes a temporal memory map used for decision-making in associative learning.
In conclusion, we found what we call decision prediction time cells. And these cells respond at different discrete times after the stimulus is presented when the animal is in the go-no-go task. In the future, we'd like to find out whether these are decision-making time cells, meaning these are cells whose activity is important for the animal. And we'll study that, but actually studying holographic optogenetic stimulation of the CA1 cells to turn them on or off.